So Axiom is striving to use AI to provide high quality safety data, cheaper, faster, and paired with deeper mechanistic understanding than is available through in vitro or in vivo toxicity assessment. We're beginning with the liver because that's responsible for the lion's share of drug failures in the clinic, in the market, and in discovery. And then our plan is to expand to other tissues in next year, including skin, heart, kidney, etc. Best models come from the best data. So we've begun by building a massive and really diverse data set of human liver biology. We're screening primary human hepatocytes with 5 pooled donors, and we're multiplexing cell viability and cell death readouts with cell painting to measure changes in the organelles, to image changes to cell organelles, including the DNA, the mitochondria, the RNA, the cytoskeleton, the ER. We've profiled 40,000 molecules so far, and this is climbing fast. And as we just heard, it's really important and useful to have full dose response curves. So we're profiling all of these across a full 8 concentration dose response curve. The compounds that we're screening, we began with all of the commercially available compounds that have been in the clinic that we could get. And we've been augmenting that with a very diverse set of both drug-like molecules, so kinase inhibitors, macrocycles, molecular glues, protacs, and more, and also molecules that are interesting to other industries, agrochemicals, natural products, cosmetics. The goal is for this to be a very diverse data set of all of the small molecules, bioactive small molecules that might be interesting across industries. And cell painting lets us measure morphological changes that occur long before cell death and that can be implicated in downstream DILI. So for example, the cell painting will pick up on mitochondrial damage, on endoplasmic reticulum stress, changes to lipids, which can be indicated in fibrosis and microsteatosis, and formation of reactive oxygen species. We can also detect changes happening to the hepatocyte long before cell death occurs. So here we have an example where cell death is beginning to occur at 11 micromolar, but we model whether a cell culture is distinguishable from the cell cultures you get with vehicle control. And here at 0.4 micromolar, there's a bioactive point of departure and the cell culture becomes distinguishable from vehicle control. So we generate full dose response curves for all of our cytotoxicity readouts, all of our biological phenotype readouts, and for this bioactivity readout. And often we see the bioactivity readout or some of the phenotype readouts have the IC50 or IC20 1 to 2 orders of magnitude earlier in terms of dose than the cytotoxicity readouts. And these bioactivity readouts display diverse phenotypes. So here we see bioactive perturbations, which have a very low bioactive dose coming from formation of vacuoles, from reduction of lipids, and from cytoskeleton disorganization and mitochondrial fission. So there are 3 main packages that we're offering right now. One is liver cytotoxicity. So this all, of course, operates just on the molecular structure or SMILE string, doesn't require synthesis. For liver cytotoxicity, we profile metabolic viability, cell death, necrosis, and apoptosis. For the biological phenotype package, we profile mitochondrial tox, cytoplasmic vacuolation, cytoskeletal rearrangement, ceatosis, and we're adding more to this, as well as providing that bioactive point of departure, that lowest dose at which the cell becomes distinguishable from an unperturbed hepatocyte. And then we also have an investigative assistant, which highlights the substructures in a molecule, which are driving toxicity, and which can reduce toxicity if those are modified. And we also produce thousands of bioisosteric suggestions to molecules that minimally change both the overall structure and overall biological properties of the molecule while maximally reducing cytotoxicity. So our goal is for this to come in at a tiny fraction of the price of running the actual physical assay at the CRO, which of course is enabled because this is all digital. So we're starting off with the cytotoxicity and biological phenotype packages at dollars per molecule, as opposed to potentially thousands of dollars running those for a molecule at the CRO. The investigative Assistant, of course, is something you can't get for a physical assay. And then our hope is to, in 2025, also be able to provide clinical readouts such as AST and ALT elevation, and metabolism readouts such as CYP induction and metabolite profiling. So the goal here, of course, is to improve DMTA. I think we all know that whether you're developing a drug or whether you're developing a molecule for some other industry, the process of doing DMTA in a wet lab is super expensive. And so if we can do more design in silico between every wet lab cycle, then we can move that process faster and also help bring safer molecules to humans. The prediction pipeline we're using uses many different molecular fingerprints and descriptors, but then we also train graph neural networks, graph transformers. We also use 3D structural information, and we also have models that we've trained to map between chemical structure and biological phenotypes. This is very similar to the models that Google and OpenAI will train to map between language and images, except here, instead of, we're mapping between the chemical structure and the images. And then we also use those models to help us prioritize new data and as building blocks for, tackling the next readout and the next modality. So as mentioned, we predict full dose response curves for all of our readouts. And here we see plotted in yellow the dose response curves for metabolic viability and predicted metabolic viability, in gray assay metabolic viability, and they match closely for many known toxicants. We have a really high correlation with the assay across various holdout sets. This is a holdout set of the FDA DILI rank list, and we get close to 0.9 PS&R correlation. We get similarly high results across different scaffold-based splits. But more exciting than our ability to predict the outcome of the assay is the ability to predict human drug-induced liver injury. And so we compare our model apples to apples against a variety of different in vitro experiments. This is a paper from AstraZeneca and Genentech, where they compared 3D spheroids to 2D human liver cell assays at predicting downstream DILI. And the paper showed that the spheroid is more sensitive than the 2D human liver cell assay. Our model is almost as sensitive as the spheroid, while coming in at higher specificity than both the 2D and 3D assays. InSphero released a benchmark paper just a few days ago, and we immediately grabbed the data from that benchmark and plugged it into our model. We get slightly lower sensitivity than InSphero at predicting DILI, but once again we're quite close. And this is a in silico assay that costs dollars to run and doesn't require synthesis, as opposed to a micro tissue assay that has a material cost and requires you to synthesize compounds. And there's a similar story comparing to benchmarks from Roche and Pfizer, where we see that this equals or outperforms 2D cell viability assays and approaches the sensitivity of 3D spheroid assays. So we're already competitive at predicting DILI with the in vitro models, and this is the worst it's ever going to be. So our goal is to add more data and add data coming from different assay types, and for this to improve on any in vitro assay that's on the market and eventually any in vivo assay. I want to talk through briefly just a few case studies. The first is trivofloxacin. So this is an antibiotic that was brought to market by Pfizer in the late 90s. It was withdrawn due to DILI, and follow-up research showed that the mechanism of DILI is this cyclopropamine ring being oxidized by CYPs to a toxic aldehyde. Our investigative assistant highlights this cyclopropamine ring as the key substructure driving toxicity and the key substructure that should be changed to reduce it. And our bioesoteric suggestions center around modifying this ring. Another case study is alpidem and zolpidem. These are both anxiety medications developed by Sanofi. Alpidem was withdrawn due to DILI, and zolpidem is marketed as Ambien and is relatively safe. Our model predicts zolpidem to be 6-fold safer in terms of its cytotoxicity LD20. And our investigative assistant highlights the differences between zolpidem and Ambien and alpidem, these chlorine groups, which are replaced with methyl groups in zolpidem, as key substructures that are driving tox. And finally, we have a case study of garticitab, which is a compound developed by Merck. This is an ATR inhibitor which failed phase 1 earlier this year due to liver tox. A compound from Merck's lead series called tuversitab later made its way successfully through phase 1. We predict tuversitab to be between 3 and 6-fold safer, depending on the readout. And we highlight the substructures on garticitab's right-hand side, the key difference between these two molecules as driving tox. So we plotted the Merck lead series as dots here. The yellow dots are the molecules from that lead series that Merck disclosed as being more potent, so less than 20 nanomolar potency. And the blue dots are the molecules from that lead series that are less potent, greater than 20 nanomolar potency IC50. And then on the x-axis, we have our predicted cytotoxicity LD20. So on the left, lower LD20, more toxic, right, higher LD20, less toxic. So what you want is a yellow dot, a potent molecule on the right that's less toxic. Garticitab was a yellow dot on the far left. Tuversitab was a yellow dot on the far right. So we believe that if we had been helping out Merck during the lead optimization phase of this program, we could have helped them prioritize tuversitab initially, to move through to preclinical development and potentially avoided that costly failed trial. Thanks so much for listening. Love to take any questions.